Hello students, this is experiment number 8 to study the rate of respiration in flower buds, leaf tissues and germinating seeds. First write the objective of the experiment, that is to study rate of respiration in flower buds, leaf tissue and germinating seeds, and then requirements for the experiments are conical flask, cork, beaker, a twice bent glass tube, a small test tube, thread, water, potassium hydroxide. Then we need germinating seeds, flower buds and leaves. Theory of the experiment. Respiration is a catalytic process or breakdown process जिसमें कॉम्प्लेक्स ऑर्गेनिक सब्सटेंसेस ऑक्सीडाइज होकर या ब्रेक होकर सिंपलर कंपाउंड्स बनाते हैं जैसे कि कार्बन डाइऑक्साइड एंड वाटर. And the rate of respiration can be measured 1. In terms of utilization of respiratory substrate, 2. In terms of utilization of oxygen, and 3. In terms of evolution of carbon dioxide. Means कितना respiratory substrate यूज़ हो रहा है या कितनी ऑक्सीजन यूज़ हो रही है प्रोसेस में या फिर कितनी कार्बन डाइऑक्साइड इवॉल्व हो रही है. बाय दिस वी कैन कैलकुलेट द रेट ऑफ रेस्पिरेशन. ऑल्सो rate of respiration depends on internal as well as external factors. कुछ internal factors हैं like age, physiological status, types of cells and कुछ external factors हैं like temperature and availability of oxygen. They control the rate of respiration. Check the chemical reaction involved in the process. Here, one molecule of glucose when combines with 6 molecules of oxygen, it forms 6 molecules of carbon dioxide, 6 molecules of water and energy is released in the form of ATP. ATP is adenosine triphosphate and this energy is used for various activities. Now come to the procedure involved. See how to set the apparatus first. First take 10 grams of germinating seeds in the conical flask as shown in the picture. Then hang a small test tube containing potassium hydroxide solution inside the flask with the help of a thread. Next, introduce one end of the bent glass tube into the conical flask through the cork and dip the free end of the tube in a beaker containing water. Now your apparatus is set. Make the apparatus airtight. Note the initial level of water in the glass tube and leave the apparatus undisturbed for 30 minutes. Observe the level of water rise in the glass tube after 30 minutes and record it in the observation table. Repeat the experiment taking leaf tissue and flower buds also. Draw these diagrams in your practical notebook. Label them properly. You can also use pencil color to shade different parts. Then write observations in the table given. Make 3 columns and write the readings of your observation as done for your reference. Then write results. Write the rise in water level in all 3 sets of experiments for germinating seeds, for flower buds and for leaves. And it is highest in germinating seeds and lowest in case of leaves. Come to the conclusion part. The germinating seeds or flower buds or leaves respire and take oxygen from the flask and release carbon dioxide gas. And the potassium hydroxide in the test tube absorbs carbon dioxide, creating a vacuum in the flask. As a result, water rises in the glass tube. पोटेशियम हाइड्रोक्साइड सॉल्यूशन रेस्पिरेशन के दौरान जो कार्बन डाइऑक्साइड निकल रही है उसको एब्जॉर्ब कर लेता है और फ्लास्क में वैक्यूम क्रिएट होने के कारण ग्लास ट्यूब में वाटर लेवल राइज कर जाता है. नाउ राइट द इक्वेशन इन्वॉल्व्ड ड्यूरिंग दिस प्रोसेस एज गिवन बिलो. Carbon dioxide reacts with potassium hydroxide and gives potassium carbonate and water. Now, let us conclude the results also. According to the results, rise in water level is highest in germinating seeds. It is 1.5 centimeter because the metabolic activity is greatest in germinating seeds, thus have highest respiratory rate. Also, developing flower buds show high rate of respiration as water level in this setup rises up to 1.0 centimeter, but in flower buds, rate of respiration is less than germinating seeds. While in vegetative tissues like leaves, they show low rate of respiration because the metabolic activity is less in mature and differentiated tissue. So, the useful rule of thumb is that the greater the overall metabolic activity of given tissue, the higher its respiratory rate. जितना ज़्यादा मेटाबॉलिक एक्टिविटी टिश्यू में होगी उतनी ही ज़्यादा उसकी रेस्पिरेटरी रेट होगा. Now write precautions. First, the apparatus should always be kept airtight. Second, fresh potassium hydroxide should be taken for the experiment. Third, rise in water level should be accurately measured. So that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.